Hi all. Test your knowledge on Staphylococcus with this MCQ. Challenge yourself on key facts about this important gram-positive pathogen and its role in infections. This quiz covers Staphylococcus species like Staphylococcus aureus and coagulase-negative Staphylococcus, virulence factors and pathogenesis, laboratory identification and biochemical test. Like, share and subscribe for more microbiology quizzes and turn on notifications to stay updated. The first question is a very simple question. The arrangement of Staphylococci is A. Chain, B. Clusters, C. Pairs, or none of the above. The answer is clusters. Of course, you know Staphylococci are cocci arranged in clusters, whereas Streptococci are cocci arranged in chain. The next question is, oil paint appearance of Staphylococcus aureus occurs on which of these media? Is it nutrient agar, McConkey's agar, milk agar, or ludlam medium? Oil paint appearance of Staphylococcus aureus. The answer is nutrient agar. Staphylococcus aureus produces golden yellow colored colonies on nutrient agar and it produces a oil paint appearance, and on blood agar it produces beta hemolysis, that is a clear transparent zone around the colony. The next question is, scalded skin syndrome caused by Staphylococcus aureus is mediated by which of these toxins? Is it hemolysin, is it coagulase, enterotoxin, or exfoliative toxin? So staphylococcal scalded skin syndrome is a common pediatric skin infection caused by Staphylococcus aureus, and the toxin responsible for this condition is, the answer is exfoliative toxin. So you can see this is the normal skin. There are different layers of skin on which when exfoliative toxin acts, there will be exfoliation or the skin will be shedding. That is scalded skin syndrome. Next question is, co-agglutination reaction is mediated by which of these? MEC-A, protein A, coagulase, or clumping factor? So this co-agglutination occurs with Staphylococcus aureus when a specific antibody of the Staphylococcus aureus binds. There will be co-agglutination, that is when it attaches with the specific antigen, there will be co-agglutination. So which of these is responsible for co-agglutination? The answer is protein A. That is Staphylococcus aureus covan 1 strain contains certain proteins called as protein A. Their specific antibody will bind with this protein A, and when they are combined with its antigen, there will be another antigen, there will be particle agglutination, which is called as co-agglutination. So protein A is responsible for this. Next question is, Staphylococcus aureus differentiated from coagulase negative Staphylococcus by all except. The options are coagulase, protein A, catalase, or mannitol. So Staphylococcus aureus and coagulase negative Staphylococcus can be differentiated by which of these tests? Except. Exception is catalase test. It can be differentiated by all other tests like coagulase test, protein A test, and mannitol test. Because in mannitol test, you can see Staphylococcus epidermis, which is a coagulase negative Staphylococcus, appears like pink colored, and Staphylococcus aureus appears yellow colored. And coagulase A and protein A test is used for differentiation of Staphylococcus aureus and coagulase negative Staphylococci. So in Staphylococcus aureus, coagulase test as well as protein A test, they are positive, whereas it is negative in coagulase negative staphylococci. Next question is, which sugar fermentation test differentiates Staphylococcus aureus from coagulase negative Staphylococcus? Options are glucose, sucrose, lactose or mannitol. And the answer is, of course, mannitol, because as we have discussed earlier, on mannitol salt agar, Staphylococcus aureus produce yellow colonies, whereas coagulase negative Staphylococcus produce pink colonies. And E. coli gives no growth. So Staphylococcus aureus gives yellow colonies and coagulase negative Staphylococcus gives pink colonies. So mannitol can be used for differentiation of Staphylococcus aureus and coagulase negative Staphylococcus. So Staphylococcus is a genus of gram-positive bacteria in the family Staphylococcaceae from the order Bacillales. And under the microscope, they appear spherical or cocci, and they form in grape-like clusters. Thank you.